within the when we open up the tree, we see what type of data in. So if I'd like to see what's in the clinical node, well, I see that I have cell type information, whether whether the samples were collected from CD4 T cells or CD8 T cells. You have information on the disease state. So, for example, for the cohorts, healthy, non-severe, severe asthmatics in this case. The medication that they've been on, so it's all it's stored in a text format, and this is denoted by the ABC prefix. And then you have data on forced exhaled volume of air, FEV1, and forced something volume capacity, whatever the measurement is in asthma. Which these we understand that these are numerical data residing in this node because of the 1, 2, 3 prefix. So if I'd like to do a quick analysis and ask if there's any difference between the healthy individuals and the severe asthmatics, so I put them in the relevant query boxes, and I'd ask it to compare the FEV1 range. So whenever you put a numerical ID, you have the option to filter by specific values. You can get an idea of what the data distribution is by asking for the histogram, so you can see how the data is distributed specifically for the FEV1. And you also get, if I get the histogram for a subset, I understand for the subset I've selected above, this is the actual distribution. So if I wanted to make a meaningful operation, I could say, give me anything that's above greater than 100, for example, just if I was interested in this type of population. So you can imagine that this kind of function will be very useful if we're trying to subcategorize and substratify some patient cohorts based on whichever clinical characteristic we choose. So we'll put it in here. I will not filter it by any value. I'm going to take the entire data set, and I'll have to do the same here. So no value, and then I ask it to generate the summary statistics. So here, what it will do, it will give me, yeah.
Is it possible to query for to temporary query to ask for the latest value, for example? Because when I have the medical history, I will have a lot of timestamps, and I just want the highest value less than blah blah in the last 3 months or something like that, where the patient is involved in the study. Or
okay, so anyone, the room is free for if anyone wants to help me with it. So in my understanding, you can't ask for the latest, but if you've organized your data, if you've curated and loaded it within visits, and you would select the latest visit, then you would be able to find the data you're looking for. I don't know of any actual filtering or logic function that you can ask to bring out the latest values. Okay, so the breakdown of what we're seeing here, it will give you an analysis of what of the subsets that you've compared. So it'll give you a comparison of age. It'll give you some information on this, a histogram of age. It'll tell you what sexes you have distribution. If we had information about race, this would pop up here. And finally, you get your analysis result on the question you asked. So you can see that, for example, the healthy individuals compared to the severe individuals, which was based on the query I did, have a much higher FEV1 percentage, and this is tranSMART tells me as well that this is the p-value for the comparison, and this is statistically significant. So if I wanted to then export this data, so okay, fair enough, I've done my analysis, I'd like to export this data and save it. There's two ways. I can either look at it on the grid view and get all the information which were used to build the result, or and this is easily you can copy and paste this table easily, or you also have this export button as well, which you ask it to export the summary statistics. It saves it to your local drive, and then you can open it within your whatever of interest. You might want to take it away and do your analysis elsewhere.
What's the data export tab right next to the review?
Okay, so the data export tab is where you can. This is all the data within the selected cohort. So I can tell it that I would wish for for it to give me for subset 1 this data, and then what it will do is it will eventually ask it for all the data. I even ask it for the gene expression, and when I tell it to export, it will start a job, collect all the data, and once this has been, it tells you once it's been it's completed, it will tell you here it's completed and give you a link. At the moment, this specific tab is still a little bit buggy and it just needs a little bit of tweaking. So I'm guessing on the Oracle version this works. Nevertheless, the export function is there either through so you have 3 different ways, and in this way I just showed you one. I can show you then how to export some of the microarray data as well. So this is all fine, I am guessing. So you do your analysis, you see, and that you might want to save this as well. So once you save your analysis, it saves with a specific ID. Now you have to be logged on to tranSMART in order to be able to retrieve this ID. But the minute you put it in, the good thing about tranSMART is that you can email it as well. So straight away your email application will open up, and you'll have the link. The minute I can send that to Jay here, and I can tell him, Jay, check out my analysis. These are the parameters I did. What do you think? And you can also, via the ID, what we've been doing in the UBioPred is we've been saving it to their knowledge portal, which is the collaboration platform which supplements tranSMART. So anyone that's, if the clinician, Peter Sturck mentioned this yesterday, they write down the hypothesis and what question they'd wish to be answered. People come in and for the statisticians, for example, come in, do the analysis, and save the analysis results using this ID specifically somewhere to be kept. So it's very useful for collaboration purposes. It will save the comparison, so I can actually show you the minute I clicked on this this link. What it will do is it will rebuild the query. So it's rebuilt the query, and when I generate, I'll get the exact same results. No, you have to be logged on. So for this instance, this is open. This is just our test platform. But for UBioPred specifically, a user would have had to log on to the tranSMART instance via their account, and then get to be able to view the analysis. Yes.
Will this work for all the input parameters that you put?
Yeah. So if it takes whatever I've built here, whatever query I've built, this is what it rebuilds.
And what about the next steps or the advanced work?
Advanced work. We'll come to that in a minute. So yep.
Just a quick question: is the data editable as well for the end user?
No. It's already it goes through the curation process. It's inserted into tranSMART. All the you can set via the administration panel here. It's on the side. You can actually tell you can assign which users can have just read access, or they also have download access as well to the data. You can specify specific groups with specific users who can perform certain actions on.
It's always
Yes.
How long is it saved? Is there time limits?
The link.
Yeah.
There is no time limit, because what it does is saves the comparison under your username, under Fabian's username, and then, as I said, you have to, the person who's viewing the link has to also have credentials in order to come in and see this.
Storing in the client side.
Yes, No, it's on. It's not storing on the client side. It's storing on the server side. Yeah. So there's a log in the back with the administration panel, which, yeah, I can even go into the admin panel. Actually, that would be quite useful to do if I could see the end of the screen. No. Where's the admin panel? Yeah, no, this it hasn't. We haven't set up. And there is an administration panel that you have. You see that all of the users that are logged in, what comparisons they've saved. You have a full activity for the administrator to be able to control what's been happening.
So when you look at all these views, there are always two subsets.
Yes, it's only two subsets.
Yeah, in the discussion we had last Monday about the demonstrator. So yesterday morning, about the trade demonstrator. There was quite clearly a need for more than two subsets. So sometimes you want to make multiple comparisons and show them next to each other. Right? Yeah.
Okay, so since this is the use cases and the requirements gathering session, this is one of the as a sort of data manager and kind of account manager for UBioPred. This is one of the requests I've had. So we try and get all of these. We try and get all of these requests down and documented by the end of this meeting. This is going to be the purpose as well. But I agree.
So if you would send the link of your analysis to your collaborator, does that imply that, for instance, the patient cohorts that you're studying is kind of stable, or if in the meantime you would add a few patients, what would happen to the analysis? Would it be a new analysis? Would it link to the old analysis?
Very good question. It will pick up. It will select the patients that you. But if more patients have been added, which match these criteria that you've been asked for, the analysis will be different. So this is something that I think from the way that we're handling it in UBioPred is that we inform when the next database update is going to be. So all these requests are valid for the. So if you've made a request and you found a significant difference and it's 0.04%, and then the next time you rerun it, it's gone. Well, you kind of consider that it wasn't really significant in the first place.
But does it mean that if you send a link like this is the analysis, does it state somewhere this is the version of the database that it's?
No, it doesn't. It doesn't. So the only way around that will be something like a screenshot on a print to PDF of the. So you can also print the your analysis and save it. So this is the way that we produce the reports within UBioPred. So this they just print the screen, they attach it to either save it as PDF or whichever means you want. I just want to demo something else that it doesn't become apparent, especially for a data set like this, but in typical data sets, and I think talking to David as well, he will tell you that some of the studies loaded in Sanofi are very, very long and hundreds of maybe thousands of variables stored. It would be inefficient to try and build all the comparisons in this way, just to put in each time you have to put in the variables in both fields, no value filter and everything. So one of the easiest ways to do this, and this is quite interesting, I find anyway, at least save some time, is the minute I set my subsets that I'm interested in. Let's not forget I can do a different type of query here. So I can ask it to substratify patients straight away. So I can query based on FEV1, and based on the histogram I'll take below 45, below 75. And for this one, greater than 75. Just make sure that. Okay, I like to say I can get an indication of how many subsets, how many subjects are in this, match these criteria. So what I can do then is, when I go to the results analysis, if I would like to see the information on FEC, I can just drag and drop it into the pane. and it'll give me the FEC statistics as well. And I can do this as many times as I want and add variables. So I can say, okay, yeah, compare this variable and then this parameter and this clinical parameter, and then again, when I come to the grid view, it will have collected all of the information and saved it. So if you want to do multiple comparisons quite rapidly. So what last thing that I'd like to show you is what, as I said, I'm honest, this is the current Postgre functionality. This is not currently the functionality that the Oracle version has. Oracle version has much higher, and our aim is to try and build the system by the September release. Hopefully everyone's in line with that. So if I wanted to do some gene expression data, this is one of the areas which is most of the development has been in recently, to try and enable all of these different analysis and gene expression analyses in Postgre. And the people who know a little bit more about this, they know that there was a lot of problems when we migrated from Oracle, and this is what's been fixed. So for biomarker data, I'll just show you a simple way. So thanks to the Gustavo from the Hive, he managed to debug this, at least make the data exportable. So if you go into advanced workflows, I've already told it that I want to look at gene expression data. From the analysis, I'll ask it to give me the heat map. I say heat map, this is what works, and it's a way you can export all the data. So within here in the heat map variable, I need to give it the node again, and I need to tell it that this is high dimensional data. And then I ask it to run the analysis. So you may have noticed I didn't filter or anything, because this is currently still a couple of things that need fixing. But there is, as I said, at least we give the projects the functionality to be able to export their data and to retrieve it and to analyze it elsewhere. So you'll see what happens, what the result is from here.
Did you pick the genes that you want to see in the heat map?
No, I didn't. I asked it to do basically what I'm doing is a significant selection of the two cohorts. This has been hard coded, the two different subsets. It just gives me the list of significant genes. So it gives me the classical R non-eligible, non-readable heat map, which is okay. It doesn't mean much. It tells me which of the. As I said, this has been fixed recently by Gustavo, helped by Shitzai from our team as well. And one of the good things is that at least you can download the raw data, all the R data, and so here you'll find that it gives you the heat map, the PNG that was used, and then it gives you all of the different data sets. So you can hear, just by virtue of the size, and I'm not going to sit and open it now, you can see that this is the entire data set. So all of the matrix of gene expression. The output file has information about fold change of each specific probe on in the two states. So which is essentially what you need to be able to rerun the analysis anywhere else. So this is where we're at the moment. We're actively working on on getting these few bugs fixed to be able to filter by specific patients and ask. So I am only interested in the subset. What is the differences in gene expression level? And just since this is just the demo version, one of the ways that we've leveraged this in proteomics data, understanding that we can't store, tranSMART can currently doesn't store mass spectra data. What we've been doing in UBioPred is we've got the list of proteins and an identifier and the values coming from the readings. So as you would see a gene expression table. So we've leveraged this function and be able to store and export this type of data as well. And I think that's it. And any more questions?
So what you have been showing, will that all go into the version 1.1, or is that for later on?
No, this is all 1.1.
Yeah.
So what we describe this as sort of minimally functional. So any project we're serving and supporting can actually put the data in, get the data out. This is our first, the first most important thing that we try to achieve, and then we want to start with fixing all the different analytical capabilities.
The basic heat map is part of this advanced workflow.
Yes.
Are there any other functions in there yet?
No, there's. I could probably try and see if anything else is still working or has survived the migration. But I could tell you that this is the only one I know that's currently working for sure. As I said, if we had an Oracle version, we would be able to show these other functions.
The others are probably not working.
Yeah, so just to let you know, the R scripts are there. It's just that someone to tell the application that when you're looking for the data for the IC50 or the I don't know, the survival analysis, where to look in the database. So this is the link that's been broken with migration.
What does it mean by IC50?
IC50. It's IC50 is the experiment you do when you're trying to find the most potent dose of the drug.
I know, but what's the analysis behind that?
Oh, I'm not sure. I'm not sure. All of the R scripts which used are on the GitHub. If you open up the tranSMART app, if you look at R analysis tab within GitHub, sorry, R modules, yeah.
That might be as discussion of what is being done. Have this more transparently visible at the top of ICT. It was this thing that was used to determine that the user always has the possibility to go back what is presented on the
True. So a documentation, the protocol which and the algorithm which used. Should we put that down?
Yeah, is that a new one though? Is IC50 new functionality? I don't think we have that on. I don't think that's a 1.0 functionality.
Potentially.
I think it's yeah, I've not seen that one.
You run your analysis and a lot of interesting stuff comes out, and you decide that it would be nice to continue with a group of high estrogen receptor expression based on your array data. So how do you continue from there?
I don't. I take it out and I take it to my own application, and then I do at the moment anyway.
So is that where the connection to Galaxy would be a very good tool to happen easily?
Potentially, yeah.
Take it in, take it out, and use it as an iterative process? Or should in the end we aim for that basically selects from any column or any row that you could do these kind of questions and make sub-selections of your data?
So the link to Galaxy would be for example, Galaxy would be very useful to be able to carry on with the. So the way that all the development efforts are going, from what I understand, it's that we are going to try and give this capability of either connecting directly or in the intermediate, potentially provide standard data output formats which you could then use in your readily formatted. The second question, can you repeat it?
So would it be good to be able to just be able to select from any column or from any row with thresholds and get sub-selections out? Would that be something one could generate within tranSMART? If you would have your estrogen receptor data just rank from high to low, and just say, okay, from this threshold on, we make a sub-selection, and you do the same for if we only want, well, in this case it makes common sense for females, but for other markets that could be males as well.
If I understand correctly, I think this is currently possible. As I said, it's not there's a little bug in the selection, but in principle, you should be able to search from. I know that this has data in. I could ask it for high-low values, high-low flag, and that's how I'm going to separate my my patients, and I can write a series of queries to filter the patients, and then ask it for a selection to tell me what the profile would be. Yes.
You can do it on the high dimensional.
Yes, you can. So in the okay. When you put the high dimensional data in here, I think I could probably mess it up at the moment because of too many queries in. So if I put two different cohorts here, it would give me another field, a drop-down list where I can sub-select. If I've already entered the filtering criteria, it will automatically take it, from what I understand. So, for example, okay, let's do it correctly. Let's see. I don't know. Yeah. So here you can ask it. You can categorize it by. Or you can select a specific gene of interest. The query and the selection criteria is taken by the comparison box. So this would feed in automatically. As I said, it's not working at the moment.
Would you then be able to select all the genes that have an expression value higher than 0.5?
Not sure. I don't know. So you set the filtering criteria.
And the filtering here is on the gene level, so you subset the observations, the one observation list of all the growth probably. I'm not sure whether you can also specify constraints on the value you observe.
Oh, well, the significance is implicitly it means that it's above 1.5 and below 0.05 corrected p.
Can you compare between different data sets?
Okay. This is something that just comes up all the time. So here, as I showed you can actually ask for a specific pathway or gene and it will give you the values for this.
Dat is met de laatste keer best wel heel erg specifiek sinds within the study and across studies for those are different use cases.
So, when I say cross study,
But yeah, do you mean cross study or within the study? So different data sets within a study.
So I assume that each element in the tree is a data set.
yeah, if it's in the same top level node, so if you have a common node above, anything in this study can be compared. I wouldn't be able to compare the breast cancer and the asthma.
You can. I thought you could take, for example, compare the age distribution between the two studies or something like that.
It won't. It will be a separate study. This is one thing that you can't do, the cross study.
I think that's an important point to put on there. My question actually relates to that topic as well. Because if you want to work with standards, the real use case for doing that is the ability to check across studies.
Yeah, true.
So, and my specific question actually is there experience in actually using HL7 for these three data types? Because there's a great data type which might be a solution for that, but will still allow you to use the terms, whatever is used in the cohort, but it allows you to look over that data. So it allows
We're talking, is this a form of standard terminology?
where you have the data type, where you have the original text, but you also might add a term from an ontology. You might use different ontologies.
This is a very valid request, and it's come up many times. Fabian knows exactly about it for the Abbey Risk project. They're looking into this. So one of the ideas, and one of the current, at least in the Etrix perspective, we're looking into an ontology server to be able to enable across. So whether this is going to be done in collaboration with tranSMART, this is yet to be seen, but this is something I have to.
Biobanking domain, that's really the route we're trying. So that's important.
for your information, one of the wonderful things that the curation tool, with the graphical user interface developed by Sanofi called the ICE tool, Charlotte, the girl who developed it, she has entered in and it links out to different ontology repositories, and you can select which terminology you want to use, and you can map all your terms to the correct codes.
Yeah, well, that's
So this is
of
just
course the mapping. But
the mapping, but then we need to
it's there because
yeah.
that's also something.
I'm just saying this is the first step. But,
Yeah.
So just a couple things. So there is a untidy workaround for the cross-study comparisons. So if you put all of, if you need to do that, you need to do that currently. You can put your studies underneath a single node, and basically then it will allow you to search across. But there's obviously there's no rigor around how it does it, right? So you have to ensure that your fields are essentially the same and that they are comparable. The reality is that's going to be true when you implement a true cross study anyway. You don't have to do that. It's actually much more, when you actually go through the requirements of what you need to do, it's actually fairly complicated. But there is a way of doing that currently if you really need to do it. And I'm not saying it's the best solution, but there is a